My name is Andreas Kunzmann, and I'm from the Center for Tropical Marine Ecology in Bremen. I lead the ecophysiology research group there. We are currently at 20 degrees south off the coast of Namibia in the Benguela Upwelling System, and we are searching for fish. For this, we use our ring trawl, which is a net with an opening of around 1.5 square meters and a mesh size of around 500 microns. We are looking for early life stages of various fish species. Early life stages means larvae and juvenile fish. And like all living organisms, like all animals, fish need oxygen to breathe and for their metabolism. One of the things that characterizes the Benguela Upwelling System is that it has zones low in oxygen, or indeed oxygen minimum zones. They come about as the cold, nutrient-rich water comes to the surface and fuels plankton blooms, which, if they are not eaten, die off and are decomposed by bacteria. These bacteria need oxygen for the decomposition process and obtain it from the water column, leading to minimum zones which can sometimes be very extensive. A fish that has to cope with an oxygen minimum zone because it lives or feeds there or because it is carried through it by the current must develop certain adaptation mechanisms, and these are what we are especially interested in. In order to be able to carry out specific experiments with the fish, we have brought a laboratory container on board with us, which has certain equipment that enables us to simulate the low oxygen content of the upwelling system. This means we provide the fish with very specific oxygen concentrations in the water, and we observe how it copes with these oxygen conditions. Firstly, we record the metabolism in the form of oxygen consumption, and secondly, we record activity. For this, we use a video camera which records the fish around the clock for 24 to 36 hours. Here we are currently in the middle of a respiration experiment. We want to know the oxygen consumption of a fish we have here in the measurement chamber. In this case, it is a goby, which is a demersal and is known to be able to tolerate very low oxygen levels. We want to know more about how low the oxygen level can be without the fish getting very stressed. Using a computerized system, we are able to regulate the oxygen content between two limits and at the same time measure the oxygen consumption of the fish. We are keen to provide the fish with as natural conditions as possible, so we have installed a daylight lamp, which is on for 12 hours and then off for 12 hours, meaning we use a day-night rhythm for these measurements. To allow us to film uninterrupted overnight, we also have an infrared lamp, and we close this tent at night so that it is nice and dark and the fish is completely undisturbed. With the aid of the infrared light and the camera, we can just carry on filming the experiment at night and record the activity of the fish. The results of the oxygen consumption measurements so far and the tolerance give us relatively good information about what fish can tolerate, what they have adapted to, and with the help of the figures, we are able to make predictions about where we can find certain types of fish.
In addition to the physiological tolerance limits relating to low oxygen content, the oxygen consumption measurements carried out on fish larvae also tell us something about their nutritional requirements, in combination with stomach contents analyses, which tells us the dietary composition, and chemical analyses of different food organisms, which determine the different energy content. With this information, we can then convert the oxygen consumption into the daily nutritional requirements. Our results show that young life stages of various species are very different both in their physiological tolerance limits and in relation to their feeding ecology, and both are factors that can have a critical impact on the reproductive success of fish species. For example, in my doctoral thesis, I have been able to show that the larvae and young fish of horse mackerel are very resilient to external environmental influences. On the one hand, they are very tolerant of low oxygen content, but they can also successfully exploit food resources under a wide range of environmental conditions. These are two explanations as to why this species is able to reproduce successfully even under the changing environmental conditions in the North Benguela system and still represents a very important fishery resource.